today for this amazing presentation by two of the experts on this field and longtime supporters of Global Genes, Kari Rosbach and Gabrielle Rushing. It is my pleasure to welcome you both to this webinar as speakers and experts. Kari Luther Rosbach, it has more than 30 years of experience in nonprofit leadership, fundraising, and volunteer management. During Kari's tenure as president and CEO, the TSC Alliance established a comprehensive research program fostering collaboration with industry and academia to move treatments for TSC forward in a more expedited way. The organization also took an active role in educating the community to diminish time for recruitment, including pivotal trials for two FDA-approved drugs to treat TSC. And Dr. Rushing joined the TSC Alliance staff in December of 2019 and works remotely from Nashville, Tennessee. Her efforts focus on stimulating innovative research while growing the natural history database and biosample repository projects. Recently, Gabrielle launched a whole genome sequencing initiative and created the first ever self-report portal to collect patient-reported outcomes from individuals affected by tuberous sclerosis complex, TSC. She completed her PhD at Vanderbilt University, studying TSC brain tumor development and neural stem cells under the guidance of Dr. Rebecca Urhi. Apologies if I'm messing up all the names. Prior to joining TSC Alliance, Gabrielle served as project manager for the Predict Pharmacogenetic Resource for Enhanced Decisions in Care and Treatment program at Vanderbilt University Medical Center. In her free time, Gabrielle enjoys listening to live music, spending time with her dog Willow, and working out. We're so fortunate to have JS Pharmaceuticals supporting us as our webinar sponsors this time. And with that, I am passing it over to Kari and Gabrielle.
Well, thank you so much for having us. We are really thrilled to be here to talk on this topic about getting your community ready for clinical trials. It's something that we have really excelled in, I think, over the last few years and take very seriously as an organization. Sorry about that. There we go. Awesome. And excited to be with our Global Genes community. We really love working with all of you and are so grateful to be asked to present on this topic today. So first, we just want to set the stage so you know a little bit about our organization and about the disease to put context to our discussion. So first of all, the TSC Alliance was founded in 1974. That's almost 50 years ago. We are now an internationally recognized nonprofit that does everything it takes to improve the lives of those with TSC. And we do that by developing programs and resource information and support services for anybody impacted by tuberous sclerosis complex. We create and implement both public and professional awareness campaigns to raise an awareness of the disease, really to get people to a diagnosis quicker and appropriate treatment. And then the last topic, which is why we're here today, we really fund and drive research. And I'll turn it to Gabrielle.
So we use this diagram to show you that TSC affects nearly every single organ throughout life. It's really beneficial to show you guys this because as you look around, you can see, oh, these brain manifestations also appear in this disease, or this organ's also affected in this type of cancer. So the research that we do in TSC is very broadly applicable to other diseases, not only rare diseases, but bigger ones like cancer. And TSC is the leading genetic cause of both autism and epilepsy. So as you go through, you can see that people with TSC get heart tumors, kidney tumors, brain tumors, oral tumors, eye tumors, and so forth and so on. And you can see on the right-hand side some examples of a very large brain tumor. You can imagine how disastrous that can be as it continues to grow. As well as on the bottom, you can see some of the facial tumors that show up. So even if everything else is managed to a point that a person can, live without fear of dying, you can see that there are other things that affect mental health and the way they are perceived and have self-confidence in the world. So there's also a neuropsychiatric component called TAND, or TSC-associated neuropsychiatric disorders. So we wanted to put this up to really frame and show you everything about TSC as we move forward with our discussion. So Kari's going to show you how we've managed to progress our programs from the very beginning and how much time it has taken to get to where we are today.
Thank you for that, Gabrielle. We do want to start with this overview because it's taken us almost 50 years to get where we are today. We have done that over time in a way that's sustainable for our organization. And I think that's what we really want to impart to all of you. It's taking those steps no matter where you are, and it's okay if it takes a while to get there. I think the important thing is when you start a program, it's sustainable. So when we first were founded in 1974, we really spent the first 10 years building our community. Our research was really driven by our scientific advisory board. And in the 1990s is when we were just really focused on the discovery of the genes associated with TSC, which are on the 9th and 16th chromosome. Our first research awards actually weren't given until 1984. So really, as I said, the first 10 years were focused on building community. I think one of the most important things that our organization did was always put patients first and insert their voice in everything that we have done, including our very first consensus conference. So with the consensus conference in 1998, we had patients at the table, not only physicians saying kind of what were the types of care that they were giving to individuals with TSC, but patients about how it impacted them. We had our very first national TSC conference, our large patient conference that was held in 1999. And then in 2001, we relaunched a whole volunteer outreach program that established volunteer chapters across the country, really got us involved in government relations through the Department of Defense, where we helped launch a TSC research program. And you'll notice throughout my presentation, we continue to survey our community about what's important to them. And as a result, you'll see how research really was brought along during that time. In 2002, there was a pivotal research conference that the TSC Alliance helped organize, where the links between the TSC genes and the mTOR pathway were presented. From there were 10 TSC clinical trials that launched, but they were successful because we had engaged and prepared our community. In 2006, we also launched the world's first natural TSC natural history database that Gabrielle will talk about a little bit more. It still exists today and has ongoing data for over 15 years. As we went through the next decade, we of course did another constituent survey because we're always reaching back and finding out what's important to our community. But in 2012, we, with a set of 5 TSC Centers of Excellence, we started our Clinical Research Consortium. And among the first studies that they got funding from the NIH to do was an autism biomarker study and an epilepsy biomarker study. They also were awarded a Rare Disease Clinical Research Network Award from NCATS. And that, of course, led to a very pivotal clinical trial in 2016 called the PREVENT trial, because we'd actually discovered a biomarker that would tell us which newborns with TSC were likely to develop seizures. Then we launched the first preventative clinical trial for epilepsy in the United States, which, as you can imagine, trying to recruit babies before the onset of seizures really took involving our entire community. From there, as you can see, the TSC Alliance launched the biosample repository, our preclinical consortium, which actually tests in animal models, new compounds or repurposed compounds. Again, involving our community, we had an externally led patient-focused drug development meeting in 2017, another constituent survey in 2018. We launched a research business plan, bringing together this amazing research platform that we had built over 45 years. And really said, how can we take this platform to the next level to really engage our community and ultimately lead to even more clinical trials for TSC. We updated our clinical consensus guidelines in 2021, and that paper was released again last August. So there have actually been 3 clinical consensus conferences in our history. We're really proud that our first drugs were actually approved in 2010 and 2012. That was an mTOR inhibitor used to shrink brain tumors and kidney tumors. In 2020, the approval of Epidiolex to treat seizures associated with TSC. And actually on April 4th, the announcement that the FDA had approved the first topical mTOR inhibitor for the use in TSC. And then in 2021, also, we launched the second preventative clinical trial for epilepsy, this time actually using mTOR inhibitors. So instead of using an anti-seizure medication, this year we're really trying to get at the underlying genetic pathway. So as you can see, at every step of the way, the most important thing we have done is to engage our TSC community in research. And I'll turn it to Gabrielle.
Perfect timing. So as you can see, we've developed lots of programs over the year, and they come together collaboratively to catalyze research forward. So I'm going to start on the left, where you see we have an innovative research, awesome light bulb icon. And what this does, our research grants, it permits early stage, really exploratory, potentially high risk studies in the form of small focused pilot projects, as you will. And the goal is for these grants to help investigators develop preliminary data such that they can then apply for larger amounts of funding from NIH, DOD, et cetera. And I have to put a plug in. If TSC Alliance did not have this, I would not be in this role today. I actually did my PhD starting from a small pilot grant from the TSC Alliance, which spurred my whole trajectory here. So one small grant can make a really big difference for your organization and your community. And the Preclinical Consortium, which you'll see right to the right of that, is really for drug testing. It facilitates translation of the most basic mechanistic research and then develops evidence to support clinical trials and new treatments for TSC. So there's a lot of collaboration here, not only with academic investigators, but industry partners to test these compounds in not only mouse models of TSC, but other various models. You'll see in the middle, which is the Clinical Research Consortium, and that really was developed, as Corey said, as a platform to accelerate clinical trials, to reduce that recruitment time and help facilitate the advancements. It was established originally in 2012 by only 5 TSC clinics along with the Alliance, and early financial support by us helped us to establish the Data Coordinating Center and enabled coordinators to work full-time to really focus on this. And then my babies are on the right. So you'll see the biosample repository. These biosamples, such as blood, DNA, tissue samples, are connected to clinical information in the natural history database, which houses de-identified or protected information about people with TSC and the symptoms that they experience. It also includes genetic genotyping results if available. We currently have just over 2,400 people in the database itself. And I'll go into the biosample repository a little bit later in more detail, but I just want to say a major benefit to all of these research programs is that they are scalable to your available resources and then demand. So one year we might have increased need for biosamples for researchers, but then the next year we might need to pivot some of that money if a clinical trial is ready to go. So it's all scalable. I'll shift it to Kari for the next slide to speak about some of the help in these programs.
Thank you, Gabrielle. Again, we want to emphasize the sustainability of these programs. So as you build them, take on what your organization can do. We didn't build this overnight. Again, it took us about 45 years to build these resources. Oh boy, 1 second. There we go. All right. So it starts by hiring the right team. And though we have a really robust science and medical team, it started by hiring the right chief scientific officer in 2011. Today, to implement that amazing platform that Gabrielle just talked about, we actually have 3 PhDs, a very talented scientist, a staff member with a ton of clinical trial experience, and a nurse practitioner that actually worked in one of our TSC centers of excellence. So she understood from the clinic perspective what it takes to implement a clinical trial. All of these pieces together have really put us in an amazing position to drive clinical trials forward. So one of the questions I know that came in advance is how do you educate or how do you recruit for clinical trials? And the perspective that we have always taken at the TSC Alliance is that we educate our community. We educate them through webinars with key opinion leaders where we share what is the science that led to this clinical trial. Why is it important for our community? What are the risk benefits? And then have a very robust Q&A that is moderated by a member of our science and medical team that really allows individuals to ask those very important questions of our key opinion leaders. When we first started doing clinical trials, we also felt it was really important to educate our community, like, what is a clinical trial? What's phase 1, 2, and 3? What is double-blind, placebo-controlled? All of those terminology that the community really needs to know to ask the right questions. So we developed videos and we developed brochures that really explain all of those things. When we are educating about a new clinical trial, we use all of our digital platforms to promote and explain what those clinical trials are. What are the ages of the individuals that are being recruited for those clinical trials? And we do that through social media. We'll do that through blast emails. We'll do that through e-newsletters. We'll do that through our Perspective magazine, which we publish twice a year. We also know that our community leaders are our greatest ambassadors in social media or when the community comes to them to ask questions. So whenever we launch a new clinical trial, the first thing we do is have a webinar with our community leaders so that when questions are posted on social media, they'll be able to direct those individuals to the right resources or the people that are accepting applications for the clinical trials. So that is a really important part of how we educate. And they are truly on the front lines with our families. We also hold town halls. We, in our early clinical trials, we actually held town halls in person across the country when we were able to do that in as many locations as possible. So we have 36 volunteer branches of our organization, and over 2 years, when we were launching our early clinical trials, we had a town hall in every one of those locations with our TSC clinics providing that key opinion leader to present the clinical trial. And then we really try to educate, promote, and have information available when we do our World TSC Conference, which is held every 4 years, or when we do our community special events, such as our Step Forward to Cure TSC walk, run, rides, whether they're virtual or they're in person, we always try to have information on our clinical trials. And Gabrielle, anything you'd like to join in?
Yeah, I do just want to say, last year I got a little innovative as I was spending a lot of time on the telephone consenting people for a project, which is awesome. I want people to participate, but it's upwards of an hour per call. So I actually applied this whole webinar concept and translated it to an e-webinar consent process where I literally do the entire consent via slides to a broad group of people. And then if they decide they want to participate, they follow up with me and do the paperwork. So it saves me many hours by doing this. So I encourage you, if you're just getting started, to consider adding that to your regulatory protocols for approval.
Thank you for that. Excellent. And I know how effective that's been for you.
Yeah. Oh, perfect. It's me anyway. So to dive into a little bit more about the project I'm responsible for, the Natural History Database and Biosample Repository really are resources for our researchers, not only to design experiments, but also advance TSC research towards clinical trials. So I did mention earlier, it houses clinical information. So I have a little more detail here on this slide, but symptoms that present in all the major organs, and it's connected to the biological samples. So the same randomly generated ID number we create for them to de-identify them in our database is on the same biosamples, so we can connect these together. So you'll see on the right a map, and the little grayish bluish circles are our original natural history database sites, and then the teal push pins are sites that are current sites donating biosamples to us, mostly blood samples, sometimes cheek swabs, and sometimes tissues that are left over from surgeries. And then you'll see the purple push pins are the PREVENT trial that Kari mentioned, and the orange are the Rare Disease Clinical Research Network trials. And these are on here on this slide because we've actually partnered with the Clinical Research Consortium sites as an opportunity to obtain additional biosamples from these studies. So what we do is we collect blood and cheek swabs from these individuals as well, and we can connect their clinical data with their clinical trial ID number for future use. So the database itself was originally designed as a way for clinics to identify participants eligible for clinical trials, based on what symptoms do they have or what medications are they on, but really it's evolved now. We're tracking what clinical trials people are in, we're linking it to these biosamples, and we've established a self-report portal that we can also link to the same ID number to really provide researchers with this plethora of information. So it's really come a long way. Kari, you can go to the next slide. And then I have to show you guys this. Last year at the end of 2021, we reached a milestone of 2,000 samples. I think we're at 2,050 now because I just checked yesterday and that's the number in my head. So we collect these and we store them at our awesome collaborator, Van Andel Research Institute. They collect everything for us. And we distribute these to TSC researchers free of charge. Not just academic researchers, but industry can request data and samples. We just charge them the shipping fee to send it out for the samples. And any request for data or samples does go through our steering committee that we have, which has clinicians, TSC researchers, community members. Right now, it's 2 parents of younger children with TSC, but I'm happy to announce next month we are adding an individual and adult with TSC onto our committee. So you can see in the graph I'm showing you from when we first actually started collecting samples, even though the biosample repository was conceptualized and initiated in 2014, the first samples started coming in 2016. So again, it takes time, guys. You'll see the nice trajectory here. And I do want to highlight, so I joined at the very end of 2019 here. You can see we've doubled the sample count in half the time as before. So it really speaks to having someone dedicated to the initiative to really spearhead this and get the sample numbers you need. The colors are just the source. You'll see something says mobile. We initiated right when I joined a mobile phlebotomy initiative. So we don't necessarily have to rely on clinic sites to get our samples. I can send a phlebotomist right to someone's home to collect samples, which is very convenient and has really expanded our access to allow people that want to participate in research have that opportunity. And then the pie chart on the right just shows you our samples by type that we have collected so far. So you will see the majority is blood from our natural history database sites or mobile, which are merged together in this pie chart. We've gotten blood from our clinical studies. We've gotten buccal cell DNA, TASRN DNA. TASRN is the TSC Autism Center of Excellence study. So we got some DNA from them, 389 samples worth, which is exciting. And then of course we have tissues, which when I joined was, I think, less than 20 tissue samples. So we have come a long way in the past 2.5 years. Car, did you want to add something? I saw you unmuted.
Oh, no,
Oh,
just,
okay.
I just want to reiterate how much this has grown as a resource since Gabrielle has joined. Her creativity, her ability to drive the collection has made a significant difference.
Thank you. And so the next slide, I'm going to show you some engagement, ways we have engaged with the community to make sure, one, that they know about the project, two, that they know they don't have to be at a TSC clinic, and three, just provide a testimonial that if people are willing. So we added some testimonials to our webpage, and I'm showing you a screenshot on the left. It's actually a carousel, so I can continue adding people that would like to be featured and share their story. And there's quotes here, which I won't read, but you can see people are very motivated to participate and help us out, and are really glad we're doing this. Some of the ways we've gotten more people involved since I've joined are not only from the testimonials on social media or our webpage, but on all of our registration forms now for our webinars or for our events, any surveys, we've added a question about participating in the biosample repository. So the first question says, are you interested in learning more? And then the second question asks for permission to reach out to them. And then I get a list, and then I do a broad reach out via email. Usually it results in some replies, so we're continuing going. And then the e-webinar consent I did mention on a couple slides ago. I'm just showing you here a flyer that Dan Klein, one of our communications team members, helped me to make, and I think it looks wonderful. So I'm doing another one next week. If you're curious as to how this goes, you are welcome to join. Feel free to email me to the link. It will not be recorded, though, because of the nature of the webinar. It is a consent process, but I'm happy to share insights onto that. You can go to the next slide. Perfect. And then, of course, you know, we can do all this, but unless people are using these samples and these data, it's not going to go anywhere. So I did just want to highlight some really good examples of impact of our program. So on the top left, you'll see we had a 2020 publication utilizing our brain tissue from Angelique Bourdais' lab, and they found this marker, newer marker that can be a potential target for TSC-associated epilepsy. On the bottom, Marinus Pharmaceuticals, one of our industry partners, utilized some of our plasma samples in the biosample repository in a pilot study, and this gave them the confidence to move forward with their phase 2 clinical trial for ganaxolone, specifically for epilepsy and TSC. And their data were, oh, this is an old slide, but their data were released earlier this year. And they've now decided, because of the results of that phase 2, they're moving on to phase 3. So this is a direct application of just having samples on hand and sharing it with a company that they now have the confidence to continue working in TSC. We do have another company that we're working with right now using blood samples to test a point of care measurement device for drug levels. And then on the right, we have a unique seed grant program that provides a small amount of money, up to $20,000, to utilize biosamples. So you have 3 recipients from last year looking at mTOR targets in the brain, biomarkers for epilepsy along with the kidney tumors, and then looking at gene expression in brain tumors. So we've got quite a variety of research being done, and it's really exciting to see this progress. On our webpage, we do have publications that have come out of database data use, and we expect to see more biosample publications come out soon. So although we started collecting in 2016, distribution of these samples really didn't happen until 2018. So we're just now starting to see some of these papers. And you can go to the next slide. And then I've hinted at some pilot projects, as you heard in my bio at the beginning. I'm so pumped about these. One is a whole genome sequencing project. As many of you may be aware, sometimes, you know, genetic tests come back negative and someone has a clinical diagnosis of a disease, which happens quite frequently in TSC. You know, about 15% of people originally are identified as no mutation identified, or NMI. And it's very frustrating for families, as you can imagine. So in order to help spur further research, as well as give something back to the families, we got some pilot funding, thanks to Julian Gangoli, to do whole genome sequencing, wherein we had the DNA from Van Andel. We sent it to the Translational Genomics Research Institute, who did a wonderful job sequencing the entire genome. And if there were results, we actually sent the results to GeneDx to confirm it clinically, so we could have a CLIA validated report, offer those results back to the participants who we sampled, and offer them a free genetic counseling session with our partners at Memorial Hermann Texas Medical Center. So we have had 9 genetic counseling sessions so far from our original pilot of 20 people sampled, and a couple that are waiting. And I'm excited to report we'll be doing 40 more samples this year. On the right, you'll see just some text. We did launch a self-report portal, which is linked to that natural history database. It was launched the very end of 2021, I mean December 30th launched. So as of May 5th, we had 44 participants enrolled with a 70% completion rate, and I'm really excited to see this keep moving forward. And you can skip to the next slide, Corey. So this is a little bit more information about the portal and what we're actually collecting. So you heard me mention TAND, or TSC associated neuropsychiatric disorders, which is really just an umbrella term for everything that we can't quantify, you know, in a clinical setting to date. Behaviors, anxiety, learning disabilities, it all kind of falls under this neuropsychiatric umbrella. So we're partnering with a group called Tandem, which is really looking to find a way to help people who experience TAND. And we've launched the self-report quantified TAND checklist, wherein people can answer questions specifically about them or their child, if they're answering for them, about what symptoms they experience and how bothersome that symptom is. So it's not a yes or no, we're now quantitating it. And the aim of this is to actually provide unique resources to families. So depending on which symptoms they enter as highest, to provide guidance after that they can take to their doctors, and as well as potentially one day using this as an instrument in a clinical trial as an outcome measure to see, okay, does this intervention help TAND in any way? So very exciting progress moving forward here.
I love this program because, again, it involves the patient voice in getting us ready for potential future clinical trials and building a resource because of patients that will help with FDA approval in the future.
Perfect. And I created my own timeline just to show you for this project, you know, even though we had some, you know, big milestones on Kari's timeline, which is already very crowded, I just wanted to show you a little bit more detail. So, you know, we had a plan back in 2003, not me, but the organization. And in 2004, Steve Sparagana, a clinician at Texas Scottish Rite, got an award to initiate the database. So we helped with project development in 2005. Launched at only 2 clinic sites in 2006, and now we're upwards of 18 and potentially adding 4 more this year, so that's huge. 100 people were enrolled in 2007. We actually didn't do a data poll for anybody until 2009. And then in 2012, engaging with industry, they gave us support to expand our variable collection list to get data that they needed that we could collect for them. And this was also the year where we had the first publication with database data. 2014, we had a database upgrade. So we actually shifted to a new software platform that's been very great to work with. And the contracts were signed for the biosample repository. I mentioned we started collection in 2016. By 2017, we had those 18 natural history database collection sites. 2018 was our first distribution. 2019, we launched the mobile phlebotomy. And then in 2020, despite the pandemic, we had over 1,500 biosamples collected and over 2,200 people in the database, which is really exciting because I told you we ended with 2,000 samples at the end of 2021, and we launched our whole genome sequencing pilot. So this year, we launched our self-report portal, and we have 4 new projected clinic sites. So again, just to show you, timing and sustainability.
It's a process.
It's a process. So I do want to touch on briefly our Preclinical Consortium, which is led by Dean Agar on our team, VP of Translational Research. He's done a wonderful job to really spearhead collaboration with many different industry partners. So really this encourages hypothesis development, you know, potential high risk, low reward studies for drug testing, and standardized models that we house with our contract research organization and standardized assays. So you can see the timeline here. It's really, you have an idea, hypothesis in scientific terms. We can use cell-based models, including human cell models, We can do animal models, both tumor and epilepsy, as well as this year, we're hopefully finishing our TSC-associated neuropsychiatric disorder mouse model that has some similar autistic traits to what we see in humans, and then transfer hopefully to TSC Clinical Consortium. So we do have a goal to advance at least 2 projects tested, within the Preclinical Consortium to clinical trials in partnership by 2025. And just to wrap up with preclinical, we've helped position 2 therapeutics to move forward to clinical trials so far, and we are aiming for way more. We have had 17 different industry members partner with us here since 2016, 60 different compounds tested. And we have the mouse models, like I said, for not only epilepsy, but tumors and autism-related behaviors. So some ongoing, really exciting clinical translation that we can't tell too much about, as you can see, but we have a partnership with a company to move a MEK inhibitor into a clinical trial for an investigative new device this year. And MEK is a very similar pathway to mTOR. And then we have another company committed to an IND as well later this year for TSC-associated epilepsy. So we are still working hard behind the scenes.
And I think this just demonstrates the importance of collaboration and partnership. The first company that you talked about, that compound was actually suggested by an academic. And the second was a company that worked with us almost from the beginning of our preclinical consortium. So I think that just shows the importance of a patient advocacy organization helping fund and drive research in collaboration with academics and industry. So on this next slide, Gabrielle did a great job of outlining all of the different push pins that are associated here. The TSC Alliance, we realize, is extremely fortunate to have 70 TSC clinics across the country. Of those, 13 are centers of excellence. And they're centers of excellence because they're involved in clinical research or the natural history database, and they meet above and beyond standards. And so we just wanted to talk a little bit more about those studies I mentioned at the beginning. First in epilepsy is the EEG biomarker study and the anti-epileptic strategies for epilepsy. So this was testing newborns with serial EEGs before the onset of clinical seizures. And what we found, and this is published, is there is a certain spike and wave pattern that was seen in newborns before clinical seizures. That gave us a window to intervene that led to the PREVENT trial. So preventing epilepsy utilizing vigabatrin in infants with TSC, and now the STEPS 2 trial, which is utilizing sirolimus or an mTOR inhibitor to prevent the onset of seizures. For autism, we had a very long, extensive early biomarker study for autism and TSC and an autism spectrum disorder, intellectual disability and determinants in TSC. So that work is laying the amazing foundation for what we hope will be interventional studies in the future. Anything else you'd like to add to that, Gabrielle?
Oh, that was great.
All this to say that it takes all of these pieces together over time in a sustainable way to really move drug development forward in our disease and hopefully in all of yours. I think you have the great fortune that technology has really just blown forward in the not so distant past. And that allows many organizations working today in drug discovery to do it in a much more expedited way. I'll just say, always remember the patient and the patient community and that engagement, which is really helps drive the gears, like in this slide. And none of this would be possible without our amazing team at the TSC Alliance, from our science and medical team, who are at the bottom of this slide, to our community programs team, which are just above that. They're the people that really keep the pulse on our community, to our fundraising and development team, our communications and accounting team, and our executive team, which is at the top. So that is our presentation today, and we are happy to answer any questions that anybody may have.
We have lots of questions that have come along, so we've been gathering them, and I'll start in the order that came through. And if anyone has more questions, please feel free to include them in the chat, and we'll go through them in the order we've received them. So I'll
Great.
start with the ones we got live today and then go backwards to the ones that were sent during registration. Could you please elaborate on the role of the one additional scientist on staff, his or her area of expertise?
Oh, I did put in the chat. So Zoe Fuchs is our science project manager. She had formal lab training as a research assistant, not only in oncology research, but in drug delivery research, which is super applicable. But I think anyone really with formal scientific training and that critical thinking experiment mindset would be beneficial to an organization.
Could you also address how to pay for the salary for chief scientists and all the PhDs? Are there grants?
Oh, so we have done, again, over time, we hired all of these people. In 2011, the TSC Alliance launched our first research campaign for fundraising called Unlock the Cure. Since 2011, we've raised over 17 million, and that has allowed us to build our staff and to build our programs.
Okay, the next question. When creating clinical trial education materials, did you explore need IRB approval? Did you create the language documents in-house with your staff or hire out?
So I can touch on this one. So it's both, right? So for our projects internally, I do all of our regulatory materials. It is a beast. So, you know, all of our flyers for recruitment, all the email templates that I send out. So yes, we do that. But any clinical trial partnerships, typically what has to happen is for that company, they have to get whatever they're going to say approved, whatever flyer we're going to share out approved. So it does rely on the company to do that specifically for clinical trials. And then we take that and share it. So we're actually doing a webinar next week with a company, and they just sent me their IRB approval because it does take time. Kari, do you have anything to add on?
No, just sometimes we're asked to comment on the whole protocol before it's submitted, which is really the best because we can find things that we know will be challenging for our community. And that's when you know you have a really great relationship with a company.
Or wording, you know, for how to word things, because you'd be surprised. The way things are phrased sometimes can be taken the wrong way. So the earlier that you can partner with the company, the better. I do agree, Kari.
Okay, we are just starting to educate our community on clinical trials, as we have 2 starting now. Can we provide links to some of you and or Global Genes resources? If so, would you like to be acknowledged? So I can answer at least on behalf of Global Genes. We would love for you to share our resources as long as they're just downloaded and or printed with our logo on them, and they're not altered, then that is what it's there for.
Same here. Our website is an open tool for anybody that wants to come and to utilize our resources and share them. We definitely just ask that you acknowledge our creation of them.
Any
guidance on how to develop industry partners?
Yeah, I mean, I'll broadly say, and I'm going to let Kari, because she's been doing this for years and years, have resources. You know, people come to us because we have data, we have samples, we have this community engagement. We have a Preclinical Consortium. So we are attracting them. So instead of, you know, saying, okay, what can industry do for us? You have to actually flip the switch and say, what can we do for them to make them want to work in our disease? Kari, any other?
Yeah, and I'd agree. It can start really simply because we started with a natural history database, and that alone attracted industry. You don't have to have the robust platform that TSC Alliance has today, but you can start simply in a way that is meaningful, not only for your community, but to attract industry.
What are the most important things for companies and small biotechs to understand when working with patient advocacy organizations?
Well, I think something that's always imperative, right, is that they understand your disease and the broader application. So I really hit that mark, and it's an easy argument with TSC, right, because we hit so many things with mTOR pathway. mTOR pathways in all of our cells. We couldn't live without it. It's actually, for those of you who like to work out like me, it's how your muscles literally grow. You can't live without it. So having these connections to things that may be already more developed than where you're at, I think shows the utility and is more, for lack of a better word, attractive to them. But Corey, I'll let you jump in.
Yeah, I would say not only engaging with patient advocacy organizations very, very early on in the development pipeline, but to work through the patient advocacy organizations to understand the journey of those living with your disease, or in our case, with TSC. It helps really frame and shape the development pipeline, because you're really meeting the unmet needs of the patient population. And I think really understanding that, taking the time on the front end to have those meaningful conversations will make a tremendous difference when you get to clinical trials.
Second to last question. Thoughts on best practices in recruitment for trials? What should families watch out for, and questions to ask?
So first things I think is, spreading your data distribution out. So social media is amazing. Most of us are on it, but it doesn't hit everybody, right? So making sure you still use email, making sure you're highlighting it in a webinar or an open forum, such as the town halls that Kari described, to allow people to get more information, distributing it out to your clinicians so they have it in the office. So if people are just off the interwebs, putting it in there. And then we also have twice per year, look at me, I'm so prepared, these perspective magazines that go out to people's houses, hard copy, that's why I have them. So depending on timing, you can put information in here with contact information. But Kari, I'll let you elaborate.
Well, I think you did a great job from the family perspective and what questions they should ask for clinical trials. Look heavily at the risk benefits. How is this really going to impact your family? Many clinical trials require travel. And maybe one of the things we didn't touch on, Gabrielle, is we've been extremely fortunate to have some donors and through a partnership actually with NORD to have some travel dollars. So we have travel funding for families to participate in clinical trials or to access clinical care. And that's really important. And those are the things to really think about from your family's perspective, is the time and expense it takes to participate in clinical trials.
And something new this year, you just reminded me, as it's spearheaded, we are doing our world conference this year. And since I joined the staff, we are now having a research room at the conference where we can have clinical trial partners be there to have Q&A, or academic researchers be present, so that if people want to engage with research, they go to a very specific room. And we're also doing a biosample collection, blood collection on site this year, which has taken some planning. So, just getting creative, think outside the box. You don't have to do it the same way that this organization did it, or historically we've done it this way. I am the champion of pushing buttons, Kari will tell you, to be creative and try things out.
And I'd also say, Parvati, whenever your community is together, like at a World TSC conference, when you have an engaged audience, in our case of 1,000 people attending, think creatively about ways to engage them in research, whether it's a research room, in this case, recruiting for the natural history database or biosample repository. Those are true opportunities. And we always highlight research, clinical trials, and advances made in these meetings as well, that really incentivizes and excites and engages our community.
Awesome. And I know that the next question is not super simple or easy, but it's one thing that we've seen a recurring theme is there's a lot of pressure when the organization is new, but they're running against time. They feel like they're running against time, whether it's a life-limiting illness or there's a lot of, oh my God, when is it going to happen? It's a little harder to do when it's in that clinical trial pathway, because it's not as simple as if we did all of these steps, it's still going to be faster. Kari, from being so experienced in the community for this long as an organization leader, how do they keep that going? How do they not give up, but also understand that they have to explain to the community that this is a process?
Yeah, I think, research does take time, and you want to take that time to do it right. What you can do is do it wrong and then have detrimental consequences. So I think taking the time to do it right and highlighting all of the wins along the way, I think that's super important. Those triumphs, getting a registry started, getting your first 100 patients in there, having publications that come out of that. Those are all ways that people can see demonstrated progress. I think those are important to highlight.
And last question, I promise. Are there other TSC organizations? Can you touch on collaboration and prevention of duplication of efforts?
Yes. So right now in the US, we are the only national organization. We have worked very hard to keep our community whole in that way. There was a previous organization called Be Careful. We collaborated a lot. In fact, Be Careful was the first organization that had travel funds, and then we merged together. There are also 32 different associations for TSC all around the world. We actually established a loose collaboration called TSC International, and we worked together on projects. There was a publication that ran along the most recent clinical consensus conference publication called Beyond the Guidelines, where our TSC International organizations worked together to create that publication. Anything you would add, Gabrielle?
Just that we're always trying to engage further with our international partners. I get people on my eWebinar consensus that email me about participating in research, which, as you can imagine, it's a little more difficult to do that, but I have been able to open it up to send, in certain cases, cheek swabs out so they can participate in that way. Blood just simply will not get here in time. And if they have medical records, sending them to me directly so I or my research staff can do data entry. So again, I'm going to emphasize being creative, follow the regulatory laws and things like that, but get creative, try things out. That's how science works. Science doesn't operate on the time that we wish it would, but you have to try, otherwise you'll never know.
Thank you both so very much. I know our community appreciates it, and with all the questions that we have had, for patiently answering them and truly just being supportive, not just for Global Genes, but for everyone in the community. We greatly appreciate your time. And thank you to the audience for joining us bright and early this morning or evening, depending on the part of the world that you're joining us from. And we hope you have a great weekend. Thank you so much.
Thanks so much for having us.